Hello, welcome to the part on cells and structure. What we're going to do with this section, I'll do 3 videos, one with the eukaryotic cell, one with the prokaryotic and the plant cell, and then one with the movements through cell membranes. So what we're going to start with, and to be honest, I probably, I don't determine the schedule, but I probably would have done this first, and then maybe this week we do the whole energy conversion, that kind of thing. But let me start here first. And I'll pick this one. Was it this one or this one? Basically, over the years, this is one of the things. This is called the classification levels for organisms. And it actually goes in domain, which there's 3 domains. You can see this. But then it goes kingdom, phylum, class, order, family, genus, species. So when you find a new organism, the first thing you do is you figure out where you're going to place it. What domain is it going to go under? And then you start breaking it down from there. I did want to show you this. See the bacteria, spirochetes, cyanobacteria, all gram-positives. You see all the different categories here. Archaea is kind of a algae, photosynthetic algae, weird organisms. I see halophiles there. Halophiles are those are organisms that can survive extreme temperature. High salt. High osmotic pressure environments, which we do talk about that in that section. Sorry, I didn't mean to say a thermophile is the high temperature, but halophiles are high salt concentration type of organisms. And then here's, it's actually pronounced Eukarya. That has a different, this is Eukaryota, but the cell is called Eukarya. But here they are. This is a better diagram. I wanted to show you all the branches, but see, this is what we're under. And eukaryotic cells are cells with a nucleus. So that's where we're going to start. Here are the prokaryotic cells under the bacteria, and like I said, the archaea is a different, kind of a weird, little different species. So we will start here first. Now, in biology, you always cover the cell. That's part of a biology course. I wanted to do this even though we're not doing human beings just by themselves, but this part right here, a human being is about 75 trillion cells. And for every 1 cell that a human being has, we have 5 bacteria cells. So 75 trillion times 5. That's how much bacteria exist on our bodies, around the organs, inside, outside, everything. Yeah, cells are measured in microns or micrometers. That's a little thing if you ever see that. Differentiation is a term that all cells are specialized. If you're talking about a human being that we're multicellular, our cells kind of start out as like a stem cell, and then they can become any particular cell in their category, depending on hormones and places in the body and all that. But differentiation is a term for specialized. A cardiac cell is specialized for the heart. A nerve cell is specialized for the nervous system, and go on, on and on. And they do vary in their size and shape. So I'm going to go back here. Now look, just to prevent me bouncing back and forth, I gave you this picture that's under the models. And this is a eukaryotic cell. So I will be going back and forth and just kind of quickly go through here. And what we're going to do first, well, all cells are composed of organelles. Now we'll do the main part in the middle first, and then we'll do all these here on the outside. So starting off, the nucleus. And like I said, this domain, eukaryota, or eukarya, I've always heard it. Those are cells with a nucleus. And you can see there's fungi, animals, plants, looks like chromatids, flagellates, little single-celled organisms. Any cell with a nucleus is under that domain. So here's the nucleus. Now, and there was actually a question on your first quiz about this. It kind of snuck in there on me. Something about DNA. Now, DNA is found in the nucleus. And in eukaryotic cells, you can't see DNA until a cell is about to divide. And it's actually composed of what are called chromatin fibers that are dissolved in the nucleus. Now, I'm going to blow this up a little larger, but you can kind of see all the purple mixed in. Chromatin fibers are long strands of DNA. And human beings, we have 46 chromosomes. And we do actually look a little bit of chromosome number later. But you can't see chromosomes. They're not really chromosomes till a cell is about to divide. So when the cell is in its normal function, normal state, you can't see the DNA, but the DNA is found inside the nucleus. And so the nucleus controls all cell activities. DNA is a blueprint, and the cell reads that and then performs whatever functions are on that particular section or strand. And every cell has a very specific function, and the DNA determines that. So, yeah, they are spherical. They're found DNA found in what are called chromatin fibers. And I don't believe, I don't, but anyway, they're found inside the nucleus. So overall function of the nucleus is it controls all cell activities. It's like the big brain of a cell. It controls everything. Now, the nucleolus is that little dense dot. You see it labeled right here on the inside. So that guy right there is the nucleolus. It's made of RNA and protein, and it is a site for ribosome formation. Right here. So, and this is one of those things, so like, well, okay, what's a ribosome? And I'm going to bounce over to my picture. These little red dots, and there are also red dots that are stuck to this area here. This is called the endoplasmic reticulum. Great word. This is the rough endoplasmic reticulum. But see the red dots? Those are ribosomes. Ribosomes are protein factories. They synthesize proteins. What are proteins? Hormones, enzymes, the stuff we talked about last week, things that break down food and power energy conversion, parts of cells, all kinds of things. So ribosomes are very important. There's a whole group of medications that human beings take if they have a bacteria infection that are ribosomal agents. They target the bacterial cell ribosome, they wipe them out, and then the cell can't function because it can't make enzymes, it can't convert energy, it can't do anything. So that's what the nucleolus is. So here's the nucleus, and that little dense area there is the nucleolus. Nuclear membrane. Now we're going to talk about two membranes. One of these I'll have a video for you. But the nuclear membrane is also called the nuclear envelope sometimes. I'll just stick with this term. What it is, it's basically two layers of what are called phospholipids. So we're talking about this part here. See that little double layered, and it's got the little junctions here, but this thing with the pores in it, this is called the nuclear membrane. The nuclear membrane separates the nucleus and the nucleolus from the rest of the cell. You do not want everything that... Most cells just bring anything across. Anything. Viruses, toxins, whatever. They'll bring them across. You don't want those kind of things getting inside the nucleus. Just like you wouldn't want everything in your blood to get inside your brain tissue. Human beings have a barrier called the blood-brain barrier, a little tightly packed layer of capillaries. It's very hard to cross, and the goal is to prevent what's in the blood from getting inside brain tissue, because there are things in the blood you don't necessarily want all the time. So that's the nuclear membrane. It's called a selectively permeable membrane. That means that it's going to allow certain structures to cross over, certain substances to cross over, and then also leave, and then it's going to actually exclude others. So it's called selectively permeable. Not every single thing inside of a cell or leaving the nucleus can cross the nuclear membrane. What I was going to say, and there are all kinds of pictures I could have picked, but here is what is called a phospholipid. And the nuclear membrane are made of these, and also the cell membrane. We'll get to in a minute. You see polar head, nonpolar tails. This is called the phosphate part. And these two tails are fatty acids. So it's called a phospholipid. And here is the two-layered look. See, the heads are this way on each end, and then the tails are in the middle. And we're going to get to this in a minute. But this part is soluble in water. The polar head is called hydrophilic. That's the term for it. I'll show you that in a minute. And the tails are called hydrophobic. They are not soluble in water. And this whole polar, non-polar tail thing, I may have mentioned this, water molecules, the way they're configured, electrons get pulled over to one side. So water is a polar molecule, has a positive end and a negative end. Anything that is non-polar repels water. Okay? You can buy stuff called hydrophobic sand that you can pour it into water, it will not get wet. Okay, it repels the positives. It's like trying to put two sides of a magnet, the opposite polarity together. So that's the design, and we'll talk about this more with the cell membrane. But that's what makes it selectively permeable. The fact that there is a phosphate head that is permeable to water, and the non-polar two fatty acids that are not permeable to water, and that allows it to be selectively permeable. Yeah, this is cytoplasm. We'll get to all that. Okay, but that's just remember this: the nuclear membrane is a selectively permeable membrane. Allows certain things to cross over, certain things cannot. Now the organelles. So I'll go through these with you pretty quickly. We're talking about all of these. These are called membrane-bound organelles. All these colors. This is actually a mitochondria. We've already talked about these. This part, the little red dots, the parts on the inside here, this thing looks like it's got sunbeams coming off. These are centrioles. There's the cell membrane, the opening to the outside of the cell. So we'll go through those and look, the definitions are here, so there's nothing for me to add, and you can go back and read through these. But we'll start here first, the endoplasmic reticulum. Overall structure, a series of elongated canals and fluid-filled vesicles, and it travels throughout the cell. This is like the UPS system. What it does is it delivers substances around the inside of a cell. And this is supposed to represent all this blue here. See these little fluid vesicles here, all of this, including this part up here. All of this is the endoplasmic reticulum. And anything that moves inside of a cell, it's highly organized. Its job is to move that around. And the very end of this is connected to this thing called a Golgi apparatus. And you can see things are leaving. This is being secreted. But that's the endoplasmic reticulum. There's two parts to this. You got the red dotted part, which when the electron microscope was first sort of built, discovered, and utilized, when they were looking at cells, they noticed that this area looked kind of speckled or rough. And this area over here did not look speckled or rough. So all this here with the red dots, which are ribosomes, this is called the rough endoplasmic reticulum. So it has a side function. It also, because it's got ribosomes, it's also involved with what's called protein synthesis. That area over here, which is kind of, the way they have that presented is kind of weird, but you can see no dots. This is called the smooth endoplasmic reticulum. And it also has a side function. It's involved with lipids. It's lipid synthesis, and because parts, cells need that. And so that's the endoplasmic reticulum. So the rough ER has ribosomes attached. The smooth does not. And it's just a little organelle, like it says here. Transport substances around the cell interior, keep substances being transported, organized. Mitochondria. We went through this last week. That's what these little Tylenol-looking things are. These are cut in half, like this cell diagram here. See, this part's all been cut off. That's a whole mitochondria in the back. You know, this part of the nucleus has been cut off. But those little bins, remember, that's where the electron transport chain is found, the Krebs cycle. So mitochondria, 7th grade definition, they are called the powerhouse of a cell. So elongated fluid-filled sacs that have enzymes for energy conversion. And we did that last week. It's when we talked about the Krebs cycle, the electron transport chain. Mitochondria take oxygen from the atmosphere and convert food energy into ATP. So enzymes convert food energy and ATP using oxygen in the process. Remember, oxygen is the last electron acceptor. No oxygen, no energy conversion. It is called the powerhouse of a cell. A lysosome, now that could be, it's most likely this little guy on the corner, any of these little round spherical structures. These are actually called vesicles, so there's something else, but see this one here with the dark on the inside? That's a lysosome. They are called the garbage disposal. What they do, they have powerful digestive enzymes that break down substances inside of a cell, either worn cell parts, viruses, bacteria, microorganisms, food particles, anything. They break that down. I believe last week I had shown a video of a neutrophil, a white cell, chasing down some bacteria. White blood cells, there's 2 kinds of those. They eat other substances they don't recognize, and the lysosomes would then break those down. So they're called the garbage pail. Centrioles, and we're going to talk about those again later. So the yellow here, those are centrioles. Every cell has 2 pairs. So you got this pair here, there's going to be another pair. And what they do is they line up on each side of the nucleus during cell division. And at some point, they actually pull the nucleus apart. That occurs during what's called mitosis and meiosis, but they're involved with cell division. So they distribute chromosomes during cell division. Going to talk more about these later. Cilia and flagella. So this is a little protozoan. See all the cilia, they're kind of short. These are flagella. And this one actually has some little pilly things coming off, but looks like a bacteria. So movements, mainly movements. Cilia actually move a substance across the cell surface. Flagella move the entire cell. So the whole thing. The only, and this picture doesn't have it, there's no cilia here. We do have, as human beings, your respiratory tract is ciliated. It's to trap all the dirt and everything you breathe in. And then sperm have a tail. The only human cell that has a flagella is a sperm. So overall, it is movement. It says hollow-barreled organelles that are found on each side of the nucleus. I'm sorry, wrong definition. Motile extensions of the cell membrane. Yeah, human cells actually have, you have a tissue type in the lungs called pseudostratified ciliated columnar. And the whole job is to trap, they're sticky, the little hairs are sticky, and they trap what dirt, stuff that's been, when you breathe in, traps it, keeps it out of the lungs. And then, movement of the entire cell. Okay, ribosomes. Little red dots here. And we're going to talk about these more later too. There's something called transcription and translation. And translation occurs at the ribosome. But the DNA is found in the nucleus. What ends up happening is the DNA is copied, and there's a section of the copy. It's called RNA, ribonucleic acid. Kind of talked about that. But it's actually called messenger RNA. It moves out, attaches to a ribosome, and the ribosome makes the protein along the gene sequence. So for right now, they're involved with protein synthesis, and we're going to talk more about these later, but they're very important. All these organelles are important. Ribosomes are probably in the top 3. If you don't have those, you can't function. Can't function with really any of these, but like this lysosome, and I'm going to move on, but there is a very rare disease. It's called a severe immunodeficiency syndrome, where people do not have lysosomes, so they can't fight infection. Everything else works fine, but their white blood cells and everything can't break down foreign pathogens, and so they had to basically live in a sterile environment. So last two things, and then we'll stop this video. So the cytoplasm is the jelly. See how all these organelles are suspended, kind of floating? Yeah, cytoplasm is made of something called cytosol. It's a protein. It's kind of a clear jelly. If you poke the outside of a cell, it would spill out. And of course, cells are tiny. A red blood cell in human beings is 7 microns, which is tiny. But yeah, it actually holds all these organelles in place. It has a lot of the amino acids inside that the cell needs. It does give the cell some shape. So it's got some pretty important functions, but all biochemistry occurs in the cytoplasm, absolutely. So it's just a clear jelly that's holding everything together. All right, the cell membrane. Now, I was going to show you, I'm going to make sure this is turned down. There we go. Last thing on the eukaryotic cells, I had already mentioned this. The cell membrane is a double layer of phospholipids. Separates the interior of a cell from the exterior environment. I apologize. Should have had one of these pulled up already. If you blow up, that's a pretty good one. See that? If you blow a cell membrane up, see here's the 2 layers of phospholipids. The polar heads, which are hydrophilic, and then here are the nonpolar fatty acid tails, which are hydrophobic. Hydrophilic means water-loving. Hydrophobic means water-hating. These are channels that are in the cell membrane. There are certain substances that cannot cross a cell membrane. Why? Because the cell membrane is selectively permeable. And so they have to go through a channel to get either from the outside of the cell to the inside and vice versa. Yeah, hydrophilic, the phosphate part, the little head, and then hydrophobic are the fatty acid tails. Acts as a selectively permeable membrane. And why can it do that? It's because of the design. Because it's 2 layers of phospholipids, one part of that being the phosphate, which is water soluble, and then the other part being fatty acid tail, which are not soluble. Or you could even say the phosphate is polar, the fatty acid tails are nonpolar. So that design allows certain things to cross. Water can actually cross, believe it or not, the cell membrane because the phosphate head is the first part it comes in contact with. But there are certain things that are not soluble. These yellow dots, that's actually cholesterol. There's cholesterol embedded in cell membranes. Hormones in your body that are synthesized from cholesterol, like steroids, they can actually just cross right over. Testosterone, estrogen, things like that, they can cross cell membranes. Other hormones cannot. Other hormones have to bind to the outside and cause changes on the inside. This one's okay, but you can see here still, here's the 2 layers here. There's a channel, it says cell membrane receptor proteins. And look, the cell membrane, if you're in a biochemistry course, is highly complicated. It's not our purpose here, but yeah, see these little chains coming off. Some of these are glycoproteins, glycolipids. There's some cholesterol. There's these channels, I keep calling them. There's the polar heads, the nonpolar tails, but that design, the fact that it has a polar head and a nonpolar tail, and there's 2 layers of that allows it to be selectively permeable. So last thing on the cell membrane. Now this actual video is on one of the PowerPoints, the one that actually came from Park on your from your textbook. So here we're skimming up on a cell membrane. See, there's the polar heads and the nonpolar tails, and see they're showing it there that it's flexible. And see, even if you push down to the point where it becomes disorganized, it'll reorganize again like that. But that design allows it to be selectively permeable. Some things can cross over, some things cannot. And you can see there's the two fatty acid tails, there's the cholesterol. See all the little things that are coming off, and there are receptors here as well. Not really showing those. Here are the channels that certain substances have to cross over. We'll talk about that, see how they're going through. Those are things that are not permeable to the cell membrane. And that's what it would look like when you're not looking at a cross section. Yeah, it's called the fluid mosaic model. Those little heads float around everywhere and they kind of go back and forth. That looks like that's kind of like a receptor. It might be another channel, but anyway, that design allows it to be selectively permeable. The fact that it has a polar head and a nonpolar tail, or hydrophobic tails and hydrophilic heads. All right, well, I hope that helps, and that's the end of this video. I'm going to start right here with the next one.